All right, everybody. Today we are going to be covering chemical communication at the synapse and the influence of specific neurotransmitters. I'm also going to talk to you about some of the agonists and antagonists for the various neurotransmitters we're going to talk about because it is just darn interesting. So let's move on, and I want you to get out your copy here. I would print this if I were you. Maybe pause the video and go and print this if you haven't already, and that way you can do some labeling as we label the parts of the synapse. As you come to the magnified level at the area of the synapse, you may recall from the previous video that I taught you about the one that's sending the message being the presynaptic neuron and the one that is receiving the message being the postsynaptic neuron. In other words, pre before the synapse, post after the synapse. The pre one is doing the sending, the postsynaptic is doing the receiving. In the previous video, this would be a terminal button and this would be a surface of a dendrite. So let's do some labeling. Here, when the two of these come together, it used to be thought that they were touching each other and that the message was like an electrical current going through a wire. It would just continue on to the next neuron. But then it was discovered that there's actually a tiny space or gap that rests between the two neurons that are communicating at this area of the synapse. And this is called the synaptic gap. Sometimes it's called the synaptic cleft, but it is a little space between the two neurons at the point, at this junction point of the synapse. So here's your terminal button, and here's the little gap, and here's the one receiving the message. Inside the terminal button are these little spherical packets of neurotransmitter, and these are called synaptic vesicles. And here you see there are a number of them, and packaged within them are these little tiny kind of shapes that I've drawn in, and these represent the neurotransmitters. You see them up here packaged in the synaptic vesicles where they hang out until they get released. The little spherical packet will come over to the edge and fuse. When it does that, it will dump its contents of neurotransmitter into the gap. By a process then of diffusion, those neurotransmitter chemicals will come across and perhaps come into contact with the receptors on the opposite side. This is called docking when they do this. So they come across, they find a place, they dock and send their message, and then after a little while we will see they will then come back up in reuptaken into the sending area of the vesicle. And so let's do a little more labeling here. Actually, take a look. Now, notice these are a particular shape, and notice that the receptor sites are a particular shape. So these receptor sites have to be perfectly matched or perfectly shaped to fit these particular neurochemicals. So the neurotransmitters, it is said, fit like a lock and a key. And what they'll do is they will dock or plug in on the other side, send their message, and then later detach and be reuptaken into this area again. If I throw in these little circles, because I want to point out to you that if the neurochemical or other chemical that's present there is not the right shape, then it's not going to be able to dock and send a message. They have to fit just like a lock and a key. This circular shape will not fit in this receptor site or docking station. It's just not going to work. After they've sent their message, they are reuptaken back through channels in this membrane, in this terminal button, and then they are repackaged into the synaptic vesicles. And the benefit of this is it's a recycling system. So neurotransmitter chemicals are created derived from things that in your diet. And so the neuron has to work to create those. So if it can recycle some of them and reuse them, it will, because that's less effort on its part. And so they will come up, be reuptaken through these little channels, and then repackaged and then reused. Some of them will be broken down by enzymes in the gap as well. And so this is called enzymatic degradation. To degrade just means to break down. So some of them are reuptaken, some of them are degraded, some of them are broken into their parts, and then those parts are recycled. It just depends on the neurochemical itself. So that is reuptake. Now I'd like to tell you a little bit about glial cells so you can be familiar with them. Glial cells are smaller cells, supportive cells, that are also in the nervous system. Glia means glue. And so originally these were thought to be like nerve glue, that they would hold the brain and the nervous system in place, to help hold the brain cells in place. And in fact, some of them do perform this function, but there are a good number of different kinds. So there are lots and lots of these, even more than there are neurons in the brain, for example. And they tend to be smaller in size. Some are smaller than others. And they will provide structural support, that idea of them being like nerve glue. And they also provide housekeeping duties. This particular kind up here in the corner called microglia, those go around and they do a cleanup when a neuron's been damaged or dies, and so on. And one of the other things that they do, and this is why I introduced this to you, is that they produce myelin sheath. And we talked about myelin sheath previously in another video, where the myelin is the part that the fatty substance that coats the axons of neurons. And its job is to help the neurons send messages more quickly. We, in the scope of this course, don't cover exactly how the neurons communicate with the ion balance. It is covered in your book, but it's a little more detailed than we need to go into for this course. But the myelin sheath plays a role in that, in speeding up transmission in neurons. Now, myelin sheath is this fatty substance, and you see it here again. Here's a neuron, here's the cell body, here are the dendrites, here's the axon, here are your axon terminals. The message is coming from up here to down here, and this is helping to facilitate faster communication in that neuron. Now over here you see when that fatty substance is disrupted, we start having problems in the functioning of neurons. And so these are specific glial cells that wrap around these axons. Each one makes its own glial cell in certain parts of the nervous system. Sometimes there's a larger cell that has these protrusions that reaches out and myelinates different parts. It just depends on where you are in the nervous system. But they wrap around the axons, and they're going to make those impulses go quicker. And they make up something called white matter in the brain and in the nervous system, because when you go in and look at an area where a lot of axons are traveling together, you see that it looks white. And it looks white for kind of the same reason that the fat on a steak looks white. Fat just has a white color. So white matter in the brain are areas where there are lots and lots of axons passing through. And so messages are being sent from one area to another by many, many neurons. You get this white matter. Other areas of the brain are what we call gray matter. So the cortex, which we will talk about later, that's the covering on the surface of the brain. That's where we do a lot of our high-level processing. That area is gray matter, meaning it's mostly unmyelinated cell bodies. And so you have this cortical matter on the outside of the brain that's grayish in color because it's not myelinated. And then you have this more kind of central regions of the brain where messages are being passed, and those make up the white matter. This becomes relevant in a disease known as multiple sclerosis in particular, because in multiple sclerosis, which is also known as MS, what you get is the immune system starts to attack the myelin sheaths on a variety of nerves, including the motor nerves and sensory nerves. And so what you have is that there's a breakdown of function of those neurons. And so the immune system in this autoimmune disease, autoimmune means you're attacking oneself. The immune system starts to attack this myelin and break it down. And it can be kind of a continual process. For some people, it starts and then it kind of stops, and they're able to kind of pick up some function again, and then it comes back, or maybe it doesn't come back. It just depends on the type that the person has. But that's why multiple sclerosis causes problems in the visual system, it causes problems in the sensory system and in the movement system, because it starts to break down this myelin. Once an axon has been myelinated, it needs to stay myelinated to function properly. So now what we're going to do is we're going to transition a little bit and talk some about those neurochemicals that we were kind of introducing in this part here. And so psychoactive drugs and other toxins, many of them function. If they're psychoactive, they function in this system some way. So psychoactive means influencing the brain in some way, influencing your ability to think, influence your ability to move, influencing something in the nervous system. And we'll see as we go through and look at some of these chemicals that drugs can impact the system kind of in a variety of different ways. So how, for example, they might influence how these neurochemicals are synthesized or created from the components in your diet. They might influence how these are stored in these vesicles. They might influence how these are released. They might cause them to be released when they're not supposed to. They might keep them from being released when they should be. You can also have influences in receptor interaction. For example, some drugs will come in here and they'll plug into this spot. They'll come in kind of sideways, and when you consume them, they'll come in here and they'll plug in and they'll block the effects of a normal neurotransmitter. Or they'll come in sideways and they'll stimulate here. They'll plug in, and instead of just sitting there and blocking the receptor site, they'll actually stimulate, and they'll cause effects in that regard. Another way is they can challenge reuptake. And so they'll sit in here and then they'll detach, this will detach, and then they'll plug up these mechanisms that allow for reuptake, and then they'll mess with that system in some way, or how they're degraded in the gap. They'll keep them from being degraded, for example, and then they'll stay present in the gap longer. So there's all kinds of ways that drugs interfere with this system. So I need to give you a little vocabulary first. An agonist is a substance or a chemical or a drug that increases the activity of a neurotransmitter in some way. In other words, they help that neurotransmitter. They accentuate what it normally does in some way. Now, these will start to make sense as we go through some of the examples, which I think will really solidify your understanding of drug agonists versus antagonists. Now, an antagonist, if you think of an antagonist in a story, that's the one that kind of goes against the hero in a story. And so the antagonist here will decrease the activity of that neurotransmitter. In other words, it will hinder its activity in some way. So let me give you some examples. Now, this is so interesting, I think, because we're not going to cover all of these drugs, but all of these drugs act as agonists or antagonists in some way that folks are familiar with. Marijuana, cocaine, the opiate drugs, all of these drugs are working in one of these ways in the nervous system. So the first one we're going to look at is a very well-known neurochemical known as acetylcholine, and it makes muscles contract. Some people pronounce it a little bit differently. It depends on who you're looking at, but its real role is to make muscles contract. And it does other things in the nervous system too, but one of its big jobs is making muscles contract. So at the junction between a neuron and a muscle fiber is where this action is going to take place. It will release, and then you will be able to move properly. Now, if you have something, a substance that is an antagonist for this neurochemical, such as the botulinum toxin, which is the toxin that causes botulism. Now, people used to be exposed to botulism a lot more often because folks would do home canning, and they would can things, and then they wouldn't heat the materials up hot enough, they wouldn't boil them long enough, and then this toxin would be present, and then when the lid was sealed, it would reproduce, and it would, if you ate it, it could absolutely kill you. It's extremely, extremely toxic. People would get very, very sick, and most people would die when exposed to this toxin. This is also the one that shows up in honey, and so the spores of it show up in honey, and for infants this is a problem because their stomach acid can't destroy the spores, but in an adult it can destroy the spores. In a canning situation, it's beyond the spores. It's actually active at that point in time. I remember being a child and being over at my dad's house one weekend and kind of looking for through the cupboards to find something to eat, and I found an actual can, so a metal can that was puffed out all around it, all along the sides, and it was like it was going to explode, and that was likely that the canning wasn't proper, and that was botulism inside reproducing and actually forcing the can to spread out. So botulinum toxin stops the release of acetylcholine, which is abbreviated the ACH, to muscles. And so if you release acetylcholine into the gap, and then it goes to this muscle fiber and stimulates it, you have normal movement. So your arm, for example, would move back and forth normally. If you stop release to the muscle, what you get is paralysis. You'll get no movement. And so this toxin stops the release to the muscles, which includes the diaphragmatic muscles, which means that you would no longer be able to move or breathe if you're exposed to this toxin. Another example, and this is what the illustration is curare. And curare is used in South America to put on the tips of arrows to then shoot those arrows and then stun prey, basically paralyze prey, so that they can hunt better. And I think it comes, I'm not absolutely certain, I believe it comes from one of the poison frogs and their bodies. And so it occupies receptors and blocks acetylcholine. So I want you to look at this. The first one stops release to the muscles, so you get paralysis. The second one instead gets into those receptor sites and it plugs in. And so acetylcholine comes across the gap, but it can't get in. It blocks it because the receptor's already occupied. It doesn't stimulate the receptor. If it did, it would actually be an agonist. It would act like acetylcholine and accentuate its activity. But it doesn't. It just looks enough like it that it plugs in and it blocks. Let's talk about an agonist instead. So you might guess from the picture, black widow spider venom is an agonist. And how does it do it, and what's the result? So what it does is it causes continuous release of acetylcholine from the vesicles. So instead of just putting out the proper amount, it just dumps and dumps and dumps. So let's imagine this. Normal movement, you're moving your arm, normal movement. Acetylcholine is being released at the proper rate and you are moving. If we stop that, then you'll get paralysis. Well, what about if we put too much acetylcholine in there? What's going to happen? Well, as you might imagine, it's the opposite of paralysis, uncontrollable movement and spasm. This will continue for a while until the acetylcholine is actually all used up and there's no more left. And then at that point, you'll get the opposite effect, which is paralysis. because tons of acetylcholine, spasm, 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 uncontrollable movement, and then it's all gone, and now you get paralysis. I actually had a student years ago who said, "Oh yes, I've been bitten by a black widow spider." And I said, "Well, did you get spasm?" And he said, "Oh yes, I did." And that's what happened when he was first initially bit. The next neurotransmitter that's very well known, and there are lots and lots of neurotransmitters, but we just go over a few of them in this course. Dopamine, and it's involved in physical movement, arousal, and mood. Arousal means how alert you are, and thinking. So dopamine does all of these different kinds of jobs depending on where it's working in the nervous system and what kinds of dopamine receptors it is working with. So the system is actually quite elaborate. It's not just that there's one specific type of receptor, there are different types of receptors. That's why it can do all these different things in the nervous system depending on where it's working. So let's look at dopamine agonists. Remember, agonists are going to do something to kind of accentuate. So in Parkinson's disease, dopamine plays an active role. People that have Parkinson's disease have trouble with initiating movement, and they tend to get really slowed down over time. They want to move, but they can't. They may have little tremors of the body as well. And this is all linked to low dopamine levels. What they do to treat Parkinson's disease in the initial stages of the disease is they give a drug called L-DOPA. And L-DOPA is a precursor to dopamine. You can't give people dopamine directly because it's the wrong size. It won't get into the nervous system, into the brain, if you just give someone dopamine. So if you give them L-DOPA, then in the brain it will be converted into dopamine. And so L-DOPA increases the production of dopamine and thus increases the use of dopamine and in the places where it's needed. And for a time, this really helps people with Parkinson's disease. Why doesn't it work forever? The reason it doesn't work forever is because the cells in a particular area of the brain involved in Parkinson's disease are dying. And so initially you can give them a boost by giving them extra dopamine that they can use easily and quickly. But eventually enough of the cells have died that there's no cells left to utilize that dopamine that comes from L-DOPA. Dopamine antagonist. This disease, schizophrenia, is linked with excess dopamine activity. Now, schizophrenia is a very interesting and complex disorder in many ways. The other neurochemicals appear to play a role sometimes. It's very complicated, but what they have found is that schizophrenia seems to be associated with excess dopamine activity. And there's too much dopamine. And so the brain does things that are unusual. So, for example, it will perhaps produce hallucinations. Those are false perceptions, like they may see things that aren't there, hear voices that aren't there. It may also cause false beliefs, and so the person may be what we call delusional thinking. They believe things are true that are not true, and sometimes those are connected to the hallucinations actually. And so this excess dopamine activity is not something you want happening in your brain. So what they do is they give folks antipsychotic medications, and the antipsychotics block the dopamine receptors. So they come in again sideways. They come in down into the gap and they plug in. And when dopamine in excess is released, they're sitting in a lot of those receptors and blocking that dopamine from getting in. And they find that folks with these prominent symptoms get better when they're taking antipsychotic medications. And again, those are dopamine antagonists. They reduce the activity of dopamine. Let's look at serotonin. Serotonin is involved in arousal, which doesn't mean like Usually you hear arousal in common jargon when folks are talking about like sexual arousal. That's not what we're talking about here. We're talking about arousal, meaning alertness. How alert are you? How kind of awake are you? How with it are you? And mood. So there is a very well-known serotonin agonist. So this serotonin agonist is called a selective serotonin, meaning it just works on serotonin, reuptake inhibitor. And so I want you to look at this end part. So this SSRIs is what they are called. Here's the SSRI. And so I want you to look at this end part. Reuptake inhibitor. What does it do to reuptake? It stops or reduces reuptake. So normally what you get is you get in the gap, normally what you get is you get the neurotransmitters coming out of the vesicles into the gap. By the process of diffusion, they go across and they stimulate the receptors. Then they detach and they go back up into the gap and they're kind of reabsorbed or reuptaken into that sending neuron and repackaged and then reused. What happens here is that this drug kind of blocks those little reuptake channels. It gets in there and it blocks them. And so now the serotonin has come across the gap, it's done its job, and it goes back to the sending neuron and it says, knock, 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 let me in. And it says, you stay out there. And so all the while, there's more serotonin being released into the gap naturally and to do these activities. And so now there's more and more and more serotonin. It can't get back up into the sending neuron. So the density of that serotonin in the gap gets bigger and bigger and bigger. And those ones that went back and said, hey, let me in, and they were told to get lost, they go, oh, okay, well, I'll go stimulate that receptor again, I guess. And so you get a high density of serotonin in the gap, which means more stimulation of the serotonin receptors. We're going to just cruise through two more. GABA. GABA is the main inhibitory neurotransmitter chemical. And so with GABA, if you give someone, since it's inhibitory, what that means is GABA says, don't send the message. That just calms everything down. And so if you are increasing GABA in some way, what you're getting is a GABA agonist. And so tranquilizers are medications that increase GABA activity. And tranquilizers, as you may know, make people relax. And if you give them enough, it makes them sleepy. And if you give them enough, it makes them pass out. Now that's why you have to be careful with certain kinds of drugs and medications where they'll say things like, don't combine these tranquilizers with other sedatives, other ones that have similar effects in the nervous system, like alcohol, because you don't want to over suppress or inhibit the nervous system, because if you do, it will get so suppressed, it'll reduce GABA activity so much that it can kill the person. It can inhibit breathing. It can inhibit all sorts of things in the body that you need to have happening to stay alive. Now here's our last one, endorphins. Now endorphins are a collection of neurochemicals that are collectively known as endorphins. And their main job in our nervous system, when they're produced naturally, are for pain killing. They also give us a real nice feeling. The runner's high has been associated with endorphin release. Some of the activity of things like acupuncture have been associated with the activity of endorphins. So the runner's high, just to be clear, is when you may have had it before. I've had it a few times. I'm not much of a runner, but you run and run and run and run and run, and you feel like you can't go on, and everything's hurting, and you can't take one more step, and you just keep pushing yourself, and eventually you kind of feel fabulous. You feel like you're kind of running on air. You feel all good. Everything's wonderful. And that is from the release of endorphin. All that pain seems to stimulate the release of endorphin to help you keep going. So endorphin agonists, an example would be the opiate medications or opioids. Now those are going to get into the gap. They're going to come in sideways, get into the gap, and stimulate those receptors for endorphin that you already have in your brain. And they're going to get in there and stimulate and stimulate. And what do people feel? Well, it's more than you would normally feel with your body's endorphins. So people feel really pain-free. They feel a little high, relaxed, just kind of fabulous. And that is one of the reasons why we are experiencing an opioid crisis in this country, where tens of thousands of people every year are being overdosed with these medications and by accident, and they are dying from it. Because if you give too much of these drugs, it overwhelms the system. It causes the person to stop breathing and so on, and they can die from an overdose. And so this is one of the reasons that opioid medications are really strictly controlled in our country now. Even people that have chronic pain problems have trouble now getting the medications they need regularly because of this crisis with opioid medications. And so again, they're stimulating those endorphin areas, and if they stimulate them too much, then it becomes overwhelming and the body just diminishes its normal activity so much that the person dies. I hope that you have enjoyed learning a little bit about these drugs. Now, if you go and do some readings, if you're interested in other drugs that I didn't talk about, or toxins or poisons, you can certainly do a little bit of looking around and you'll be able to find their mechanism of action. If you did some searching and you put in mechanism of action, you would be able to now understand how marijuana works, how cocaine works, how other toxins like that from the pufferfish work, because these are the systems in which they're working, and they're either agonists or antagonists.